Welcome. In this podcast, we're going to delve a little bit more deeply into the analytical applications of emission spectroscopy. By the end of the podcast, students should be able to compare UV-vis and emission spectroscopy in terms of their analytical figures of merit, explain the basis for chemiluminescence, electrogenerated chemiluminescence, Forster resonance energy transfer, and green fluorescent protein, and how these tools and techniques are used in analytical science. So let's get going. First, I'd like to talk with you for a few minutes about the analytical figures of merit and begin to show you how you can use the analytical figures of merit to think about and compare different techniques. So first, if you recall, there are 5 or 6 different terms that we've talked about that are really important. The 5 that you've already heard are sensitivity, limit of detection, or LOD, accuracy, precision, and linearity. And if you recall, I also spoke to you about the importance of practical considerations. Well, there's one more figure of merit that you should also be thinking about, and that's molecular specificity. So what we're going to do is we're going to take a look at these 6 terms: sensitivity, molecular specificity, limit of detection, accuracy, precision, and linearity for emission. And what I'd like you to do is to think about them and compare and contrast these analytical figures of merit with those for UV-vis. So, compared to UV-vis, emission spectroscopy has a much higher sensitivity. In point of fact, it can actually be part per trillion if you have a very, very strong chromophore. The technique has a high molecular specificity because not all compounds that absorb, for example, UV visible light emit, and they don't all emit equally well. And so the molecular specificity of emission spectroscopy can be a very powerful tool in allowing you to discriminate between structurally similar substrates. Another strength is its limit of detection, which is actually much lower than that for UV-vis. In point of fact, solutions as dilute as 0.1 picomolar can be detected if you have a very strong fluorophore. Emission spectroscopy is highly accurate and it shows very good precision. I'd say it's comparable to UV-vis in both respects. And I should mention that calibration curves for emission spectroscopy can be prepared and may be linear over as many as 6 decades of concentration. That's again a strength of this technique. So, with these characteristics, it really shouldn't surprise you that emission spectroscopy is actually used quite widely in analytical science. And for this reason, that's why we want to take a look at some of the applications of emission spectroscopy in this short podcast. First, there are a number of, I'll call them simple techniques that are based on direct detection of emission. In particular, many drugs are strongly emissive. For example, as little as 2 parts per billion of lysergic acid, or LSD as you may know it by that acronym, can be detected using fluorescent spectroscopy. And in point of fact, emission spectroscopy is actually the FDA standard analytical technique for this particular drug. In hospitals, the analysis of many electrolytes, lipids, and many blood parameters, all of these are performed using emission spectroscopy. It's cheap. The methods tend to be robust from an analytical standpoint. We'll talk more about exactly what that means in a few podcasts. And the instrumentation it can be used by anyone, pretty much. Doesn't require a specialist. For those of you who remember CSI, the ever popular fingerprint collection and visualization technique, based on the use of superglue. That basically is based on the emission of the superglue. All of these represent direct applications of emission spectroscopy. Emission can also be used indirectly. And by this, what I'm talking about is the fact that many compounds will actually react with compounds that are emissive. And when they do, they will quench the emission signal for the emissive compound. And by quench, it means they'll decrease the intensity of that emission signal. And so you can use this principle, for example, as the basis of imaging in neurobiology. Another variation on emission that's used widely in analysis is chemiluminescence. And sometimes you'll see the acronym capital C, capital L used. Chemiluminescence is the phenomena of detecting emission that is produced from a photoexcited species that's generated as the result of a chemical reaction. And the bioluminescence of fireflies is really a great example of chemiluminescence found in nature. In chemical biology, a technique known as Forrester resonant energy transfer, which spells out FRET, FRET as it's often referred to, is now used widely in bioanalysis. The fundamental principal phenomena involves two good chromophores for which the absorption and emission spectra overlap. And you place these two chromophores physically in close proximity, and if their dipoles are aligned, then energy can be transferred between the two chromophores. The result is that you excite the receiving chromophore, and it'll produce an excited state that's emissive, and that energy can be detected. You can use the fact that these two chromophores have to be in close proximity to obtain distance information on the two chromophores, and in point of fact on anything that might be attached to them. So FRET's been used quite powerfully in bioanalysis for example, among other things, the conformational analysis of nucleic acids, proteins, the study of protein complexes. And it's also been used in imaging applications as well. Still another interesting set of applications is based on the use of so-called green fluorescent protein, or GFP. So what is GFP, or green fluorescent protein? It's a protein that was originally isolated from a jellyfish that emitted light at 509 nanometers when you excited it with blue light at 395 nanometers. And the basis of the effect is the binding of calcium ions to a photoprotein that's called aquiorin. And when the calcium binds, the resulting complex that's formed is very emissive. It's so emissive that it's almost borders on amazing. The quantum yield for emission for GFP approaches 0.8. That's really, really high. A number of different structurally modified GFPs have been developed whose emissive properties quite literally provide coverage across the visible spectral range. And I show you a set of those on this slide. In molecular and cellular biology, GFP is used as a so-called reporter in expression. And GFPs have been developed and used in a wide range of biosensors. I'd be remiss if I didn't mention that the 208 Nobel Prize in Chemistry was awarded to Osamu Shimomura, Marty Chaffee, and Roger Tsien for their discovery and development of all of the wonderful science that is the basis of green fluorescent protein in bioanalysis. And I provided for you a couple really nice references that you can check out at the bottom of the slide. I highly recommend the GFP site. Still another important analytical technique that's been developed and is now used commercially quite widely is called electrogenerated chemiluminescence, or ECL. This technique is an example of a so-called spectro-electrochemical technique. It represents the marriage of spectroscopy and electrochemistry. In ECL, light is generated through a chemical and an electrochemical process. And as I show you on the next, I think a couple slides later, there are a number of companies today offering analyzers for a number of important problems based on ECL technology. Let's start by taking a look at what I'll call the workhorse for the technique. And it's an old buddy of mine. The compound is called ruthenium tris-bipyridyl. And basically you bind 3 2,2' bipyridyls through their nitrogens to a ruthenium, and you form this highly symmetric, almost octahedral complex. And highlighted in purple on the slide are the nitrogens, which are bound to the central ruthenium colored pink in the rendering at right. We talked a bit about this compound before, and so I think you'll recall that you can excite it to form a photoexcited singlet state. The absorption transition is a metal to ligand charge transfer transition. That means the ground state HOMO is metal centered on the ruthenium, and the LUMO is centered on the bipyridyl, and it will very quickly undergo intersystem crossing to form an excited triplet state. And that triplet state is, relatively speaking, highly emissive. But just to draw a comparison with the green fluorescent protein that we just got through looking at, the quantum yield for emission for ruthenium tris-bipyridyl, which is not a natural compound, is only 0.042. Ruthenium tris-bipyridyl emits at 610 nanometers, and I've already told you its phosphorescence, its lifetime, therefore, should make sense to you at 600 nanoseconds, relatively long-lived. And it's those characteristics, the comparatively high for a synthetic material, quantum yield, the wavelength for the emission, 610, and the relatively long lifetime for that emissive state, 600 nanoseconds, again, it's because it's phosphorescence, that make it really the workhorse of this technique. So now I talk to you about how it works, and the fundamental phenomenon is referred to as annihilation. And so I want to depict for you how the process goes on this slide. In annihilation, ion annihilation, you have a radical cation, you have a radical anion, and they combine. And when they combine, they produce a photoexcited state that is emissive. In ECL, the excited state that's formed is this ruthenium tris-bipyridyl that we just talked about, that triplet state. And here on this slide, we do what folks often do and show a little asterisk to indicate the photoexcited species. Now, what we're going to do is we're going to carry out the process electrochemically. So we're going to change the voltage in solution, in a solution that contains ruthenium tris-bipyridyl, and we're going to step it both in an oxidizing sense and then in a reducing sense, so that we can oxidize the ruthenium. We can generate ruthenium tris-bipyridyl 3+, the oxidized form of rubipi 2+, and then we'll switch the potential in a reducing sense, so we'll generate near the electrode reduced ruthenium 1+. And that 1+ and 3+ species in close proximity at the electrode nearby will react with one another and ultimately produce one photoexcited molecule of ruthenium tris-bipyridyl that's emissive. Hence, we have the ability now to detect. So now, how do we do detection of something interesting? Well, on the slide at left, I show you references to a couple of the commercial instrument manufacturers, just to highlight for you the importance of this technique in the real world. It's really found two, I'll call them niches: clinical diagnostics and biodefense. And as an example of how these assays work, we'll look at an early study done in 1991 that was based around the use of tripropylamine, often referred to as TPA, and N-hydroxysuccinimide modified ruthenium trisbipyridine. And NHS ester labeling, this using N-hydroxysuccinimide, it's frequently a reagent that's used in biological chemistry to make attachments to amino groups under slightly alkaline conditions in water. And why do we want to do that? Well, amino groups are ubiquitous. They're commonly found in peptides, which are the building blocks for proteins, and important biological materials. All right, so how does it work? You have some material that's diagnostic of some health condition that you want to quantitate. If it were an antigen, you could build your assay around an antibody-antigen complex. And we've talked a bit about those before. So if we bind an antibody, that's a protein, to our label, the ruthenium trisbipyridine label, then we can use the emission of the label, the ruthenium trisbipyridine, to tell us about the antigen that's bound to our antibody. We can use the electrode to oxidize tripropylamine, and that tripropylamine will spontaneously lose a proton, and it will oxidize the ruthenium from 2+ to 3+. That TPA radical, tripropylamine radical, will then react with the oxidized ruthenium to produce a photoexcited ruthenium trisbipyridine that will emit. And we can measure the emission of the ruthenium trisbipyridine, which in essence is being an optical label, and this will serve as the basis for our assay. Since this is an optical emission assay, it shouldn't surprise you that it has very low detection limits, sub-picomolar. So let's look at another example. Say you wanted to test for a specific serotype of salmonella. You're concerned about food poisoning. You could use an appropriate O or H antigen and build your assay around an antibody-antigen complex using ECL. You could bind one antibody to a magnetic bead, magnetic so we can fish it out of solution and rinse it off any excess reagent, excuse me, and then capture the antigen. If you bind another antibody to our label, the ruthenium trisbipyridine, then we could form a sandwich. You've seen this before, and we can use the emission from the ruthenium trisbipyridine label to tell us about the antigen. And I've included some performance data on this slide to help you to see that this can be done relatively quickly, and that the assay is relatively sensitive. And I want to stress that depending on what problem you choose, what one's sense of speed is may change. So here we're talking about detecting bacteria. And so for detecting salmonella, 24-hour turnaround is actually considered to be pretty good. All right, I hope at this point you can see that there are many different advantages potentially to this particular approach, ECL. And so I want to summarize them for you on the slide. You can do this for different experimental approaches the same way you can do it for major classes of techniques. So some advantages are ECL is non-destructive, it's sensitive, we've taken a look at that. It's selective. If you don't have the fluorophore tag, then you're not going to detect it. Fluorescence is an optical tool, so it's relatively fast. And you can do this at what are called ultramicro electrodes, really tiny electrodes. And when you do it at tiny electrodes, you can do the detection as quickly as in microseconds. You can carry out your assay and look at concentrations over 4 decades of concentration. Since there are 4 major manufacturers, it should make sense to you that this is a very automatable technique. If you're talking about clinical assays or biodefense, well, you don't want to deal with radioactivity and you don't want to deal with the waste. So all of those characteristics also become strong advantages. And something else that's really cool is you don't need a lamp. So it makes your design of your instrumentation a lot simpler. It's also compatible with all kinds of complex biomatrices. So you start to think about blood or food and dairy, working with, say, milk or cottage cheese, or working with meat tissue. Very compatible. Disadvantages, you need a very pure fluorophore reagent. And that fluorophore itself, in terms of the tag, can be relatively expensive. But in spite of that, it should be very clear you have quite a few advantages, and that's the reason you have so many companies that have developed automated, dedicated ECL instruments. I think you might be interested in looking into this technique a little bit more, and so that's my only purpose in providing you with these references on this slide. Alan J. Bard, who was a former editor for Journal of the American Chemical Society and a professor at University Texas Austin, is the developer of the technique, and so you might find his monograph quite interesting. I've also provided you with a relatively new review article from JES and 2 classic articles on ECL. And that will bring our discussion of ECL and applications of emission spectroscopy to a close. And as usual, I hope you'll take a moment to reflect before you leave the podcast and ask yourself at this point these questions. Are you comfortable? Can you compare and contrast UV-vis and emission spectroscopy in terms of their analytical figures of merit? And what is chemiluminescence, electrogenerated chemiluminescence, FRET, and GFP? And can you discuss with others how these tools and techniques might be used in analytical science? Thanks for watching. Look forward to seeing you again.